Good morning, everyone. It's really a pleasure to be here. And I'm happy to share with you some information, maybe some reasoning behind FSMA. So we'll take a little break from the actual regulations and talk about some of the science that feeds some of those regulations. And as Wes said, I work at University of Delaware in the Department of Animal and Food Sciences. So I do research in that area as well as teach. And I'm really happy to be here with you today. So I think you're familiar with the numbers, right? About 1 in 6 Americans have foodborne illness each year. We are very fortunate and blessed to be in a country where we don't necessarily think about the risk, right? You ate breakfast this morning, maybe you had a snack already, and you didn't think about the risk when you were ingesting that food. That's definitely a privilege, right? But as you are all aware, and as we've been talking about this morning, this pre-harvest environment is full of risks. And so we have to think about, how do we mitigate? Where do we put more efforts? Where do we get more bang for our buck, so to speak, right? And so that's where we're going to kind of look today at some of the research that's going on relative to those questions. So I teach in the fall semester at University of Delaware a foodborne outbreaks investigation class. It's really fun. It's a lot of kind of like CSI. And we go through outbreak investigations. We talk about why things happen. And last fall, something that I've never experienced before, we had over a dozen outbreaks occur between the months of September and December when I was teaching this class. It has really been a phenomenal year when it comes to foodborne illness. And that's something that we all need to think about. We're hearing more about this in the news. People are more familiar with this. Is it because the organisms are there more often, or is it because we're testing, or is it because people are more aware? Maybe a combination of things. But 2015 really was a very busy year. And the Centers for Disease Control and Prevention were monitoring between 17 and 40 clusters each week throughout that year, not just within those few months that I mentioned, and over 195 multi-state clusters altogether. And that's really how these things start. You get a phone call to a physician's office, or you have more tests that are going on at a laboratory, and then the local Department of Health goes in and starts looking at it, and then there's some testing that might be involved. Then the FDA or the CDC may be called in, but certainly things happen at the state level. If we look at who the CDC was investigating last year, here you can see some select multi-state outbreaks, not all of them, but some of the bigger ones from 2015, and there are many up there, and there are many commodities, not all produce related, right? That's something certainly to keep in mind. I think a lot of times people will say produce has a target on its back, right? When there's a produce-related outbreak, it makes the news. Maybe not everything else makes the news, but certainly people are very interested in produce. It's extremely healthy. Everyone wants to eat more of it, so you hear more about it. But it's not the only one. We've got cheese and meat. We have processed food as well. So I'm going to kind of come back to this chart a little bit later, but it's been a very busy few months for investigations. And so far in 2016, we have 2 larger outbreaks that have been investigated, one involving packaged salads and Listeria monocytogenes, and another one more recent, which is involving a dried chia product. It's a meal replacement, and that has to do with Salmonella. And we're going to focus a little bit on Salmonella today because, as I mentioned, this is a dry product. Salmonella is kind of like the bully on the playground. It is the one that's going to be able to survive. It's going to be present in various environments, in many situations, ones that we don't normally think about. And so that leads me to kind of thinking about this slide. Most of the outbreaks that were just on those previous 2 slides were the perfect storm of events. Many things went wrong, or many things occurred, right? And some are controllable, some are not. Sometimes these things just happen. Maybe there's a rain event, right, that leads to sediment coming up in the irrigation pond. Maybe someone doesn't use the right amount of sanitizer. Maybe there's something that's imported that is contaminated, right? Many things may happen. But we're looking at microbial populations in our foods that are not normally supposed to be there. And that's where the scientists start questioning why. Why are these organisms on our food products when they're not supposed to be there? If we think about E. coli and Salmonella, they're very happy living in the gut, right, of animals. In particular, E. coli is very, very happy in this environment. Not so happy when it gets onto a leafy green. But why is it surviving there? And really, to mitigate that risk, what we've heard so much about this morning is we have to control water and we have to control soil amendments, because we're trying not to get the organisms that are very happy here into this type of a situation, right? So we're trying to mitigate that risk. And the science is asking questions why. Why are they surviving? Why does pathogenic E. coli, which survives very well here, now also surviving very well on a lettuce leaf? It's a very basic science question that now we're sort of being questioned by. And as Wes mentioned, there is decay in the field, and that decay is really important, right? There is a stressful environment when we're looking at produce that's growing. There's sunlight that causes UV radiation that can act on the microorganisms that are there, can result in cell death and cell desiccation. That's why that time lag is really important, right? We're also have a very competitive environment. There's a lot of bacteria that are on surfaces of plants and also in the soil, and those don't provide nice environments for bacteria, especially for E. coli. If you were to put E. coli and Salmonella kind of together and see who would be the one that would win the battle, really Salmonella is the one that's going to be able to survive more of these environments, and that's why you see it listed on a lot of the causes for outbreaks. So we have kind of those stressful situations, but then we provide niches for these organisms where there is less stress, where they may be more likely to survive. If we provide a pile of manure that doesn't reach the right temperature, that may not be turned properly, that might have a lot of moisture and nutrients, then the bacterial organisms can be very happy there. They're able to find a substrate that they can hold on to, and they're able to grow. I often, you can personify bacteria a little bit, and they are much like people. They like communities, they like friends, they need something to eat, and they need moisture. So we're trying to not give them those, right? So when we think about surface water, right, if there is a lack of nutrients that's available, that's much better for them. That's why if the sediment gets stirred up, the science shows us that those bacteria that are attached to those sediments are now coming up into the water, right? So we want to have a less stressful environment. That's why the treatment systems, or where you have less access to the environment, a covered well, those kinds of things are going to be less risk for bacterial growth and survival. So do we have kind of the perfect storm, or to ask a more scientific question, are we creating pathogens that survive better in these environments? Pathogens and bacteria have been here since the time of the dinosaurs and before, right? They are able to change their genes, they're able to grow and survive under different situations, kind of better than we are. They adapt and evolve very, very quickly, right? And that's something to keep in mind too, that as we start to put more and more hurdles in place, they are continuing to try to advance those. So we have to not give up the fight, certainly. But if we look at situations that happen on the farm, what can we do to try to minimize the risk? For example, asking questions about the water that's used for transplants. Is this a specific area of growth that we need to provide more thinking about? Do we need to use potable water here? Or do we need to use water that is of a better microbial quality when we're thinking about transplants? That's a question that's come up in the Delmarva area when thinking about tomatoes and some of the Salmonella issues that have arisen there. So I'm going to tell you about a few different research studies that University of Delaware has been a part of. Actually, for all of the studies that I'm going to mention today, there are other universities that are on board, including Rutgers, Ohio State, University of Maryland, and it's really nice now to see the USDA funding these very large projects where multi-universities are working together, so we can see what happens in the different climates, especially across the East Coast, where predominantly used to see a lot of produce research done in California and Arizona. It's really nice to know now what's happening on the East Coast as well. So the first study I'm going to show you asks the question about bacterial persistence on plants coming from irrigation water. So you have these numbers of allowable limits, and when you're testing your water, is that meaningful? And all the data I'm showing you has been given to the FDA, and actually we have the FDA working with us on a lot of these projects, and they're very invested in the science. They want to know what's going on so that the rule really reflects what's going on in the environment. So in this case, I'm going to show you some data from growing tomatoes. And we irrigated these crops with water that had very high microbial loads, lots of E. coli. And then we took samples and we looked to see how much E. coli was actually present on leaves and on the tomato fruit. And we correlated that with temperature and humidity and rain to see if that would have any effect. So here's some data from 2012, and I just want to draw your attention here. Kind of on the bottom line, we've got the amount of rainfall over this time period, and we're looking kind of at the end of August into September in 2012. And then this blue line here are numbers of E. coli, generic E. coli, and you can see they're going up here, right? So we start off with very low, basically no E. coli. Then we added this E. coli here. This is what we used in our irrigation water, kind of modeling if you were to use surface water that had quite a bit of bacteria. It drops down, so we've got decay within a couple days, right? That decay, that's really important in that stressful environment. But after it rains, we have those E. coli numbers that go up very high. And this is something that was seen year after year in Delaware and in Maryland and in Florida and in Ohio. Every time it would rain, you'd have higher counts of generic E. coli on the fruit. And so thinking about this, those kind of couple of days that you're thinking after you irrigate and you let that decay, that might also go for rainfall. Right? So this is something we're trying to tease out now, the science. Is this regrowth on the plants, or is it splash? These are tomatoes, so splash is a little bit less likely. We did this with leafy greens as well, and we saw the same thing. But when you have significant rainfalls, thinking about what might be on the bacteria populations on the plants is really important. Same thing in 2013, except for when we first started is when we had our significant rainfall. So we had about 7.8 centimeters here, and you can see a lot of regrowth. So time and time again, in multiple universities seeing the same thing, with rainfall impacting what's growing on the crops. What's interesting, though, we can take this a little step further, is look at the bacterial fingerprints. So just as if you were going to try to fingerprint in a crime to figure out who did it, we can actually take the bacterial fingerprints by looking at the DNA from the different bacteria we find on the tomatoes and see, are there the same organisms that are coming from manure that we applied and that are on the plants. And what's really interesting is that some bacteria are much stronger, are much better and able to persist in the environment than others. These might be the ones that then cause the outbreaks that we hear about. And what we did with the University of Maryland is cluster some of these based on the genes that they have and find that, yes, indeed, some of the ones that we applied did survive all 10 days. And some that just came from the environment, maybe from birds, maybe from the wind, who knows where they came from, but they were brand new, and finding them on day 7 and day 10. So right now we're trying to address what makes a better pathogen. Why do some E. coli survive and some don't? The good takeaway, though, is there is that decay, and that not all the bacteria are able to survive for those 10 days, which we looked at over the study. Okay, so that's a little bit about E. coli and generic E. coli. Let's come back to a little more interesting organism, Salmonella. And there are over 2,000 different types of Salmonella, which is why sometimes if you're listening in the media, you'll hear about Salmonella Montevideo, Salmonella Newport, Salmonella Tennessee. It seems like there's a new type of Salmonella popping up all the time. When we look at where we are seeing Salmonella, you certainly do see quite a bit associated with poultry, right, and with eggs. That's where you might be normally thinking about them, because Salmonella is a normal flora of these organisms, of animals. If you think about how much bacteria we have on our own skin, right, you know we're not as sterile, right? We certainly have a lot of different bacteria, but we won't find Salmonella on our skin. But if you went to a bird or a chicken, or if anybody, if you have children or pets at home, reptiles, amphibians, they have Salmonella as part of their normal flora, and that will contribute to what's in the environment, the Salmonella in the environment. It doesn't make them sick, but the Salmonella will make us sick, and in some cases it might make young mammals sick too, like calves might get sick from Salmonella. But these other here is kind of interesting. Anybody have any idea what that is? So we've got vine vegetables, fruits, and nuts here in the purple. Oh, it's going to tell you up here. I can't ask if it's going to give it away, right? But you've heard about sprouts already, right? We include some of the leafy greens over here. But dairy might be there as well. And there's processed foods too, right? You remember salmonella and peanut butter? Right? You've heard that. It's able to survive in these different environments. So we do see salmonella in a variety of different food products. This is sort of an interesting way to think about some of the differences between salmonella and E. coli. To kind of focus on salmonella, so those animals that I just mentioned, we've got it living in the environment. Salmonella is out there in our pre-harvest environment, and it is able to survive in the soil. It is going to decrease and decay, though, because we have sunlight. You may have variable moisture conditions. That's going to work against salmonella in some cases, right? But it is more cyclical in our environment, whereas if you look at this, which comes from a paper from 2003 by a set of scientists here, E. coli doesn't necessarily have that cyclical environment unless you get into the tropics. And that's something we need to think about when you're considering your water and your soil amendments. What's going on, and where might salmonella be generated, and where might salmonella be able to survive in your pre-harvest environment? So I think awareness is really key. So if we think about what's been happening with salmonella over time, salmonella and tomatoes kind of go hand in hand, right? It's something that we think about very often. You may also think about salmonella with cantaloupes, but today we're going to focus more on salmonella and tomatoes. And if you look historically, there are a variety of different types of salmonella, all different names here, right? And not just salmonella, we have other organisms here too. And all of these come from tomatoes. Many of them were involved in multistate outbreaks, meaning that the tomatoes were distributed throughout the country and caused illnesses in various states. But this has led to a lot of criticism and a lot of concern about how tomatoes are grown and about control. And whenever there's these large number of outbreaks, the FDA always becomes very alert, right? It becomes very concerned. And over the past couple of years, in particular in the Delmarva area, especially down in the peninsula of Virginia where we grow a lot of tomatoes, there's been a lot of concern from buyers and from the FDA and from growers as well as what we can do about this. How can we try to reduce the risk of salmonella? And then this past year, of course, now you know the joke probably that you can't spell Chipotle without E. coli, right? And there's many other outbreaks that have happened with Chipotle. They have been suffering. If you've been following the news, they actually closed down this week on February 8th to try to get every restaurant on par with their food safety. We'll see how all of that goes. But there was a case in Minneapolis and Minnesota where they thought that the tomatoes actually came from the Delmarva region. And so we were very alert again, trying to figure out what growers. In Virginia, they work very closely with this, trying to figure out what's going on. It has a lot to do, too, with that FSMA rule and where you're thinking about your distributors. This was a part of that. They weren't really sure where the tomatoes had come from, and so maybe it was their record keeping, maybe it was other issues, but of course that's been really important to Chipotle's interest as well. But it's not going away. And if we look at some of the information that comes from the Virginia Department of Health, this specific species, Newport, is ranked number 3 in isolates from human clinical symptoms. So not all salmonella are created equal. Right? Some are more likely to cause human illness than others. Now, as a grower, that doesn't necessarily make you feel better, right? And especially for some of our producers of poultry, that salmonella is involved in poultry, but a chicken may be colonized by salmonella that isn't on this list, but the USDA doesn't care. It's still salmonella. It still has that potential to cause illness. But when we look at what's happening in the pre-harvest environment for tomatoes, we're very concerned about how we can reduce the presence of this specific organism. And so there's a couple interesting bits of science that are happening and things just to keep some of your heightened awareness when you're looking at your farms. In this study in 2014, and here we are looking along the peninsula here, and this is the Delmarva area. More chickens actually up here in this area than people. In the state of Delaware, we have more chickens than people on a daily basis. And maybe Salmonella is impacted by that, but it's not necessarily the answer to it. And so here I'm going to provide you with a little bit more information. So we're looking specifically at the prevalence of that Salmonella Newport. And what they did in this study is they went out and they took various samples. They looked at landfills, and they looked at landfills where they had a lot of seagulls that were there as well. And they found that Salmonella was fairly prevalent, 17% of the samples, not in all of them though, right? And that's, I should mention, is sort of a good and a frustrating aspect of Salmonella, right? My lab group has gone out and taken lots of samples across Delaware and Maryland, looking in water, looking in sediments, looking in soil. You really don't always find it. In fact, many times you don't find it. But then the FDA might go out and look, and then they'll find it. So you never know where it's going to be, which is not very helpful, right? You can't say yes or no to certain things. But in this study, they found 22 different types of Salmonella. So it wasn't just the Salmonella Newport, but just to reinforce the fact that there are various ones out there, but they may not always lead to a public health incident. Right? And I think that's something that's important to think about. So if you've seen something like this, maybe on your farms, that of course might set up, warning bells. Not something that you want to see, but they were trying to correlate this type of a picture to the presence of Salmonella. And they were able to find that where there were birds, in particular gulls on some of these landfills, they were able to find that specific Salmonella Newport. And so they thought, well, that's a pretty important finding, because maybe we need to really specifically look at where the gulls were, maybe not some of these other animals. They did find other Salmonella. This one, Javiana, also seems to survive really well in the environment. And they looked at lots of different other sources. So some of these friends that you might have that seem to come along in the pre-harvest environment, snakes or turtles. I know we've been growing a lot of watermelons for some studies we've been doing, and I always seem to find a lot of toads and frogs there. Sometimes we pick them up and sample them, and you can find Salmonella on them. Sometimes you can't. I don't know if they are very appreciative of that. My students always enjoy that part. But here you can see there's lots of opportunity for Salmonella to be introduced into the environment, and already runoff was mentioned as well. So being alert to those types of situations, I think, is really important. Knowledge is certainly empowering in all of this. And again, if you look at this study where they were sampling from these various sources, they found variety of Salmonella. Some of the specific pattern they were looking for, that DNA fingerprint, and others that were just random and ones that they weren't expecting. And I wanted to mention here that some of amazing experts that are doing this work at Virginia Tech along the peninsula, Laura Strawn and Steve Rideout, that are other resources for you in this area. You're very lucky, I think, from Rutgers down to Virginia Tech. There's so many different experts all kind of working to provide the best science. And so if you are looking around, you'll see some of these great extension work. You'll see some really interesting studies that are going on asking about Salmonella survival as well as Listeria survival in these different conditions, what affects microbial die-off. And I hope what you'll see is that you've got this great network of scientists really all working together to try to provide the best information. How can we get more die-off in these situations so that you will be able to provide the safest product possible? And there's Laura's information as well. You can follow her and look at and see some of the interesting work that they're doing. So now to tell you a little bit about one of the studies that we've been doing looking at watermelon that relates to both E. coli and Salmonella. Watermelon is a big crop in Delaware, and so the Delaware Department of Agriculture asked us to look at this, in particular, questioning the use of poultry litter as an amendment. Certainly that's something a lot of growers are using these days, and as you heard already in FSMA, the direct application of manure to the soil is something that's in question. My lab group is actually doing a lot of work with the FDA trying to figure out that risk assessment in terms of how long should we be waiting before applying it. And the science is going to back up what comes out as we see more and more information going into that part of the rule. And that's where you've seen a lot of changes between the original and the final, certainly. So here we took some land and we applied poultry litter, we looked at both total clean out as well as cake. So when the houses are cleaned out, you have like kind of the total removal of the litter, or you might have the scraping of that top part in between flocks. And sometimes that's mixed, sometimes it's kept separate. And so we looked at both of those, applied those to the land, and then we inoculated some of the poultry litter here with a marker strain of E. coli so that we could follow it to see what was happening to that E. coli over the harvest time. And we took soil samples over 6 months, and we collected data on the presence of Salmonella, on our marker strain of E. coli, looked at the climate data, of course, kept track of the moisture and the rainfall, and now we're trying to figure out which types of Salmonella were present. So try to see if that Salmonella Newport was there, and really trying to answer a question about the use of poultry litter. And is that a higher risk, or is it not? And is there something that we should be considering? So I'll give you kind of the broad overview. We took over 1,000 soil samples, and we swabbed 120 watermelons during harvest. And in general, we were able to find the E. coli that we added to the soil as soon as day 2, and then all the way through the study, we could find it in the plots that were amended with poultry litter. Right? So, and that's something that's not necessarily a brand new scientific finding, but it correlates with what a lot of other people have been seeing, is if you have poultry litter as an amendment, it gives a substrate, it gives moisture, it gives nutrient content to the soil that allows the bacteria to be able to grow. So we were able to see our E. coli surviving there. We also identified Salmonella coming from those plots throughout the entire study. And so that's something to keep aware. And what's really interesting to me, kind of the scientific question, if we just sampled the pile here, you don't necessarily find a lot of Salmonella. But as soon as you till it into the soil, you give it the correct conditions, maybe chemically, maybe microbiologically, more moisture into the soil, and then you start to find the Salmonella, which is not necessarily the answer that growers want to know, but it's something important that we need to be able to figure out what's the risk. So while we were kind of following the Salmonella through the different soil samples, you definitely saw an increase as the weather got warmer. And perhaps this is also correlated with some wildlife. There were more flies that were coming in. There was certainly more abundance of those amphibians that I mentioned earlier. But Salmonella wasn't in all of the samples. It's in about 36% of all the soil samples. So now we're really teasing out that data to figure out what's the risk, where are the risk factors, where can we provide some information to growers on how to mitigate that risk if you're using soil amendments. When should you apply it, and when should you be harvesting? Those types of specific questions. With the watermelons, we found that 50% of the watermelons were positive for that E. coli that we added to the soil. And what's really interesting about that is you're adding the E. coli in May, and you're harvesting in September and October, and that E. coli is still surviving, right? And it's surviving the highest amounts where we had that soil amendment. And you can see there's not necessarily ground contact. You've got a lot of thick brush here. We actually grew them on plastic as well, and that E. coli was tilled into the soil. So it is surviving, right? And that speaks to kind of the persistence and the robustness of these organisms that we're looking at. And we found that Salmonella was present on some of these watermelons as well. But I want to reinforce that this doesn't necessarily set up a scenario that's a public health risk, right? You have to then have these organisms be pathogenic, have the genes to be able to make a human sick, and have to have the amount of organisms that you need to make someone sick. So while they're persistent in the environment, it doesn't necessarily directly correlate to illness. So why all this focus on Salmonella? Certainly the FDA is very interested, as well as the CDC. We've seen Salmonella survive in a variety of different food products. Again, if you go back to just these outbreaks that happened in the past few months at the end of 2015, we certainly have a resurgence in these kind of pocket pets. The CDC is very interested in following them. We could add hedgehogs to this picture as well. People are cuddling and holding them. In some states, it's actually illegal to sell sliders, these cute little turtles, because they do cause Salmonella. Children are sick by these since they're not washing their hands properly, things like that. and you see the numbers go up. More and more illnesses associated with Salmonella. And when that happens, everyone's kind of put into the pool to try to account for that, right? So we're all sort of responsible for this. Interesting, too, if you look at these outbreaks here that happened in August and September, both of these were caused by these pre-browned, pre-breaded, ready-to-eat chicken. And I know it's not a produce, but I think it's important for us to think about, again, because we're all kind of accountable when there's these increase in Salmonella. And just to reinforce that we need better education when it comes to consumers and food safety, because these products look like they're ready to eat. They're sold in the frozen section. They're sold along chicken products that are pre-browned and pre-breaded and are ready to heat instead of ready to cook. But these products actually haven't been cooked. So you have to read the labels, and maybe not everyone is really good at that, and maybe it's that food scientist dilemma of there's too many options, too many things that are available, but these continue to cause outbreaks. And so this is a very nice infographic developed by the CDC to remind people to read package instructions, and then if you have to wait the 45 minutes and cook this in the oven, then that's what you have to do, because that's what's happening with these chicken here that's not fully cooked yet. They're just pre-browned and just pre-breaded, but they look like they're ready to eat. You could just nuke them in the microwave for a few minutes and eat them, but they're not ready for that. So they may still have they need to be cooked. So they really shouldn't have Salmonella on them anyway, but they need to be cooked fully. So again, if you're looking to the consumer to try to do this, we need better education. So here we are with Salmonella. Kind of if we look over the past several years, what's happening with this line? It's not going down, right? It's not going down the way that the CDC would hope it would. We have Healthy People 2010 goals. We have Healthy People 2020 goals that are established by the Centers for Disease Control and Prevention, and we're not making them, right? And this is why there's so much focus in food safety and focus on looking at Salmonella. If we look at where we've seen Salmonella, now bringing it back, more closely related to produce, we have here our outbreak-associated hospitalizations, very high numbers of hospitalizations associated with tomatoes, right, and with fruits and nuts with Salmonella. Illnesses, again, pretty high when it comes to Salmonella with our vine stock vegetables, and unfortunately the deaths quite high as well when it comes to fruits and nuts and Salmonella. These might be associated with the people that are eating. You know, if you certainly look at some of our population that's most at risk, the very young and the very old, and those that may be on chemotherapy or may be surviving with chronic diseases. We have very large populations these days, luckily, that are able to live with chronic illnesses for longer periods of time, but they also are at risk for certain infectious diseases like Salmonella. A lower dose may make them sick. So something that we need to think about. And of course, because of these numbers, certainly then the spotlight is put back into that pre-harvest environment and what's happening when it comes to Salmonella. So just a little bit more kind of basic information. Salmonella is not the bacterial disease that you think about. I'm just going to go live in the bathroom, right? I'm going to have diarrhea, maybe a fever. I'm just not going to feel well. It's kind of, I think, what most people think about when it comes to foodborne disease. I work with norovirus quite a bit in my laboratory, and we do a lot of virus work. And so when people talk about the stomach flu, kind of, you know, makes me a little crazy because it's not. The flu is respiratory. When you think about what's happening, it's a diarrheal disease, right? So we have to try to clear up some of these misconceptions. The same thing goes with Salmonella. It's not something that you can just say, oh, I'm just going to eat my chicken raw, or it's okay if my eggs are undercooked, I'll be all right. We're seeing more and more Salmonella connected to chronic disease and illnesses like Reiter's syndrome or other types of reactive arthritis. Because what happens is you get these organisms inside your body and your immune system starts to react. It's recognizing a foreign agent, these foreign microorganisms that have really extensive proteins on the outside, and your body's reacting to it. And it may actually develop, more often now than not, these chronic diseases. There's a lot of reasons for that. Maybe it's because our immune systems are not pushed as much as they used to, right? We are a pretty clean society. Some people have that theory, but it's something to keep in mind that when you see those numbers of Salmonella, they may also correlate to these chronic and more serious illnesses. And when you ingest Salmonella, it actually gets through the stomach, that important line of defense, the acidic environment, gets into the intestine, right, can actually penetrate the cells of your intestine and go through and then become systemic, which is a very serious situation, right? You could have a systemic bacterial infection, and maybe you've heard or you've had family members or friends that have suffered with this and then have to be put on pretty extreme antibiotics to try to clear that. But Salmonella can do that, and that's why often the diarrhea that's associated with Salmonella may be bloody, may be mucousy, because you're getting that kind of tearing of the cells there. Sorry for the potty talk right before lunch. Right? Kind of comes with the territory. So I just want to reinforce that this is what's happening, kind of this cartoon diagram. You get this colonization here on your intestinal cells, and because you have all those bacteria that are there, you can't absorb the nutrients, right? So you really have to wait till those bacteria clear or go away, and then you'll be feeling better. So this kind of way that the bacteria grow is also how they may be attaching to pieces of manure or pieces of soil, because they're very good at colonizing. They're very good at attaching. And even on a lettuce leaf, for example, or on a leaf of a tomato, or on the surface of the tomato, the bacteria can kind of stack up just like this. And it does take several days to be able to figure out what's going on. So you may have cases of Salmonella that are coming in, and the local health department has notice of that, but it takes maybe a month to try to figure out where that outbreak is coming from. So just a little bit of caution there. Patience with the media and recognition, too, if you're going to be selling produce. Trace back is so important, as you are all aware, but it takes quite a long time to try to figure out exactly what's happening, because you have all the testing that's going on, and that is a very tedious process. One thing to think about is this new commodities that continue to pop up, and I'm not sure what's been happening in terms of the science behind some of these cucumber outbreaks, but we have seen cucumbers being recalled and involved in multi-state outbreaks for 3 years in a row now. And that's sort of a new finding, where it was pretty quiet until then, and now cucumbers are going to be in the spotlight. So again, something to be considering is it's not just tomatoes and cantaloupes, right, and nuts. We really have to keep thinking that Salmonella is adapting and in different environments. Some of this does come from imported produce. The 2013-2015 outbreaks were both linked to produce that was grown in Mexico and imported, but the 2014 outbreak was actually linked to cucumbers grown in Maryland. So we are not safe here, just because it's U.S. and not Mexico, and it's not necessarily just an imported issue as well, but something to think about. In fact, this is the epidemiological curve, which is how we track illnesses. We have the dates of illness here, and each little point is an individual illness. They're not additive. So on each day, we're looking at new illnesses that were popping up in the 2015 outbreak, and it went on until just recently. Actually, the CDC, just at the end of January, ended up saying that the outbreak was over. But you can see how long it continues, even with 2 recalls going on. So these are long and very tedious processes. Each outbreak that occurs when it comes to Salmonella, there is a lesson that's learned, and I think that's an important takeaway for your own water testing as well. What are you learning? What's going on your own farm? Every time something happens, we can learn a lot from that, and that happens with the outbreaks as well. Here's, again, an infographic from the CDC showing some of the more famous historical outbreaks that have happened. This was a very big one, reinforcing sanitation in 1994. We had raw eggs that were transported in a tanker truck, made into ice cream, and put back into the tanker truck without proper sanitation. Thousands of people were sick. You can see 200,000 people there that were sick because of that, reinforcing the basic need for sanitation, right? You could say, oh, it's 1994. We learned our lesson. But we still today see issues, right, with proper sanitation, even with hand washing. So just reinforcing all of that. Again, this consumption of frozen pot pies, that were not even microwaved. In that outbreak in 2007, people were just purchasing them and then eating them. Don't know about that one, but making sure we have clear instructions, right? I think everybody's sort of involved in this. We need consumers that do what they're supposed to do and follow instructions. We need companies that have clear labels and clear instructions as well. So to kind of summarize, I have just 2 more studies to show you, which again go to these differences in bacteria and the potential for introduction into the pre-harvest environment by different wildlife, by different animals. So this is a study that was done at University of Delaware, University of Florida, and University of California, where we looked at various Salmonella strains, and we over a year's time figuring out who is the one that's going to be able to persist the best. So we used a variety of different types of manure that were, in our case, collected from Delaware. California did the same from California, and Florida from Florida. What's really interesting here is that you see different survival in the different types of manure depending on what the animals are eating as well, which is kind of interesting. Again, going to the complexity of the situation. So a variety of different types of manure here. And if we look at just over the first month, we see Salmonella that's surviving really well in cattle feces and in deer, right? And that's what you might expect, because when you hear about outbreaks or you see how bacteria are being transmitted in a pre-harvest environment, those are 2 really common animals to see, right? To be thinking about might be coming in. You might have heard about this with apple cider in the past, use of drop apples, or water moving manure from cattle, those kinds of things coming into the environment. What is a little more interesting here is at least in the pigs that we had in Delaware, we saw death and decay of the organisms happening pretty quickly. Raccoon, pretty quickly as well. Sheep, kind of up and down. And waterfowl, this was our Canadian geese here, they started to decay, after about a month. So the bacteria are not surviving in all the manures the same way. And then if we went and looked at which Salmonella were surviving the best, kind of again goes by these different types of manures here over the 12 months. But you can see kind of across the board these blue bars, which is Branderupt, seems to survive the best. And Javiana survives the best too. Our Newport here, not doing so well. In fact, completely missing from 9 and 12 months, which is very interesting as well. Maybe it doesn't survive as well. So in those cases where Newport caused so many outbreaks, maybe there was a more recent contamination event. So that's something for us to be thinking about, how these organisms are getting into the environment. We did the same study looking with a variety of different types of E. coli. And just to tell you that E. coli doesn't seem to have the same specific differences that Salmonella does, but you do see this again, the survival in cattle and in waterfowl and in deer being much higher than the other animals. So the animals that you see more commonly maybe being wildlife intrusion issues in the field are the ones where both E. coli and Salmonella seem to survive better. Maybe that's because the organisms have been able to find their niche there. They're able to survive there a little bit better. And so with that, I'll take any questions, and thank you for